Early morning at St Mary's Hospital, Paddington, in London. A pathologist prepares for one of the several post-mortems he does every week. He doesn't usually take such elaborate precautions, but the patient in this body bag died of AIDS, acquired immunodeficiency syndrome, although that's not what his death certificate will say. Officially, the cause of death will be pneumonia, of a type that is a classic AIDS disease. There's also Kaposi's sarcoma, a cancer affecting his ear, lung, intestine, and feet. The pathologist's helmets protect them against two other diseases which are highly infectious. An abscess on the lung may indicate the patient had TB, and his yellow skin shows he had hepatitis B. With all those diseases, he didn't stand a chance. But even they aren't the fundamental cause of death either. One slip with the scalpel could be lethal because the blood is infected with the real killer, the thing that made his immune system collapse and left him prey to the ravages of these diseases. In one of his lymph nodes, the infection can be identified as the virus we call HIV, human immunodeficiency virus. This program is about how HIV kills, or how we think it kills. Even after a decade of research, it's a puzzle with very few pieces in place. First, how do we know that HIV really is the AIDS virus? The simple truth is that there's never been an AIDS epidemic without HIV. The memorial quilt, laid out in Washington in October 1992, commemorated 152,000 American victims of the disease. It had spread with the classic pattern of a virus, affecting groups sharing common factors, in this case, exchange of body fluids through sex and drug use. HIV also fulfills the formal scientific criteria for a virus responsible for an epidemic. For example, it can be isolated from virtually all cases of infection and be shown in laboratory cultures to kill the cells affected in AIDS. We also know from worldwide studies that most people with HIV get AIDS within 10 years, and people in similar risk categories who don't have HIV don't get AIDS. The link is inescapable. That's not to say we know how it causes AIDS, but nor is it to say there's no connection. That argument has sown a lot of doubt publicly, but almost none among scientists. Ask them whether it causes AIDS.
Yes, it does. It definitely does, but I think the confusion about that stems from the fact that the precise mechanisms whereby the virus ultimately destroys the body's immune system are not totally and completely understood. It's very clear that the virus is the primary agent involved in setting off a series of events that ultimately lead to the destruction of the immune system.
Well, there's no question in my mind that HIV by itself can cause AIDS and does cause AIDS. However, it's also clear that there are many other extenuating circumstances and other infectious agents that will accelerate the process of clinical disease development after you're infected with HIV.
Unfortunately, many members of the public have been exposed to the idea that HIV might not be the cause of AIDS, and this is clearly a most ridiculous notion. It is the overwhelming body of scientific evidence is that this virus leads to this disease, and the only dispute is basically what are the mechanisms in between getting the infection and the disease.
To begin at the beginning, a virus, any virus, consists of little more than genes and an overcoat of fats and proteins, and underneath genes made of nucleic acid, which carry the blueprint of its future existence. Unlike a bacterium, which is a single cell that can reproduce itself, a virus cannot reproduce on its own. It has to find a host cell, penetrate it, and use its chemistry to help make copies of its own genes and proteins. Then new viruses bud from the host and seek out other cells to infect in order to continue the viral line. They even make the organism do things to help them, like causing sneezing or cold sores to spread the viruses. The mechanism by which some viruses replicate is different. At one stage in their life cycle, in order to make new proteins, they convert their RNA back into its mirror image, DNA. Hence, they're called retroviruses. HIV is a retrovirus. Since the Earth cooled and life began, viruses have been evolving in concert with living cells. Because viruses are not fully independently alive, there's a chicken and egg question about which came first, virus or cell.
Well, there's a lot of controversy about how viruses actually originated. There's a lot of evidence to suggest that, as Howard Temin of the University of Wisconsin originally suggested, many viruses are actually pieces of cellular genetic material that may have escaped from the cell at some point and acquired an independent existence. So that suggests that perhaps there weren't viruses until the cells had already evolved. Now, other people think that there may have been a prebiotic world made up of RNA, and that some of the viruses existing today, perhaps influenza viruses and some of the other RNA-containing viruses, are vestiges of the prebiotic RNA world, the earliest form of life on Earth.
All forms of life are subject to viruses, even bacteria. Over the millions of years of evolution, the human body has learnt to coexist with them, so that today we all walk around with an inner baggage of viruses which may never make us ill.
For example, human beings are host to 7 different herpes viruses. And we're all familiar with various diseases caused by herpes viruses. We know about herpes simplex, for example, cold sores or fever blisters. We know about chicken pox. That's a herpes virus. It stays in our bodies for the rest of our lives, generally under control, quiescent, and unnoticed after the initial infection. But years later, many of us will get a reactivation of that early infection. The virus suddenly reawakens and causes disease, zoster or shingles, usually a mild disease. But it's a reminder that we've been carrying that virus all our lives.
Other viruses have often caused serious epidemics, though 200 years of vaccination has made a difference. This man, suffering from smallpox in 1977, was the last recorded case in the world. In the 1930s, poliomyelitis paralyzed nerves and put patients who couldn't breathe unaided into iron lungs. At the end of the First World War, a worldwide flu epidemic killed over 20 million, more than the fighting had done. The government of the day issued public service films, just as it was to do for AIDS. Not all these life-threatening viruses are human in origin. We know that flu, for example, can cross to mammals from bird species like cormorants and ducks, and then from mammals to man.
Influenza viruses really are natural infections of wild waterfowl. And ducks are really the natural library of influenza viruses. Just about any kind of influenza virus you want to look for can be found in some waterfowl somewhere in the world. How do these pandemics get started? Well, they all seem, at least those in recent history that can be traced back, to have originated in China. Why China? Well, we don't know for sure, but the most logical hypothesis is that China, interestingly, has a system that's been practiced for a long time of integrated pig-duck agriculture. Now, pigs, it turns out, can also be infected by many of the same influenza viruses that infect humans. And many people see them as a kind of mixing vessel that can allow avian or bird influenza strains to infect mammals. And of course, the farmer is right there in this pig-duck agriculture, so when the farmer comes by to tend the infected pig, he can catch influenza, and a pandemic can start.
The idea is that the same may be true of HIV, and it's microscopic analysis of the genes of certain animal viruses that gives reason to think so. Not long ago, such viruses were invisible. A tiny dot would cover about 230 million of them. But in the last 30 years or so, we've had electron microscopes powerful enough to let us see not just the viruses, but their very core. That's made it possible to classify viruses into types, such as the slow-acting lentiviruses.
In fact, the human AIDS virus is simply a member of a large family of non-human primate lentiviruses. So there are closely related viruses to the human AIDS virus which infect a wide variety of African monkey species. The African green monkey, the sooty mangabey, the mandrill, and the like are all infected with their own AIDS-like virus.
That virus is called SIV, which is simian or monkey immunodeficiency virus. It's genetically similar to HIV-2, the less virulent strain of the human AIDS virus. It's mostly found in Africa, though it does occur in Britain and the rest of Europe. It's usually accepted that this genetic similarity points to the origins of HIV.
It seems very likely to me that the HIV originated somewhere in Africa. And I say that because we know that close relatives of the HIV are still present, very widely distributed in free-living monkeys throughout sub-Saharan Africa. But such viruses are not present at all in free-living monkeys in Asia or South America, for example. So that's quite compelling evidence, I believe, that the virus originated in some subhuman primate species in sub-Saharan Africa.
Those African monkeys carry SIV without getting ill. If we knew why, it would help explain how humans could be protected from HIV.
The fact that a virus can affect an animal and make it ill, and yet when it affects a very similar animal, there is no illness, raises the question that as well as looking at the virology, the anatomy, and the physiology of the virus itself, we're also going to have to consider immunology, the study of the immune system, how the immune system responds to the virus, and how the virus affects the immune system.
To look at the human immune response is to look at one of the body's most complex systems.
The immune system can be viewed as a military operation whose job it is to defend the organism from foreign invaders. Just as that military operation might have a Navy and an Air Force and an artillery unit stationed on land, the immune system has lymphocytes that are able to make antibodies. It has lymphocytes that are able to kill invading organisms. It has macrophages, which are able to eat invading organisms.
To enter the world our virus inhabits, we have to descend deep into the human body, a fantastic voyage into another microscopic universe. Floating in the blood in our arteries are two types of blood cell, red ones that color the blood and carry oxygen, and white ones that fight disease. Some of the white blood cells are scavengers, like this macrophage, swallowing up bacteria and cell debris. Others with large nuclei are called lymphocytes and play the lead in this drama. We have 1 trillion of them, give or take, and there are two main types. The T cells are responsible for killing cells that already have virus inside them. Some of the T cells are the actual killers, some are their helpers. The other main type of lymphocyte is the B cell, which produces Y-shaped strings of protein called antibodies. They latch on to any free-floating virus in order to immobilize it, and also to attract other immune cells to come and destroy it. The defense forces of our immune system are constantly on patrol, linked by a highly sophisticated communication system. They're trying to stop any virus before it can enter a cell and occupy it. The enemy carries distinctive markings. The coating of each virus is studded with proteins, the foreign bodies called antigens. As viruses begin to invade the bloodstream, some of them are consumed by the frontline defenders. Other cells chop up the antigens and display bits of them on their surfaces. Then they call in helper T cells, already programmed to recognize these specific foreign bodies. This encounter activates the T cells. They send urgent chemical signals to stimulate more helper and killer cells to multiply. Another signal stimulates the growth of B cells. As they grow in numbers, within hours another signal tells them to start producing antibodies, those proteins which can block the virus. Meanwhile, some of the viruses have found their targets and entered the cells of the body. By now, the killer T cells have been activated and are on a search and destroy mission. They sacrifice infected cells by chemically puncturing their membranes, killing the virus inside. A cascade of signals amplifies the immune response until the enemy is overwhelmed by sheer force of numbers. Memory cells are left in circulation, which will recognize the infection if it ever returns and trigger the same response. It is these memory cells which a vaccine creates, without first causing the disease. What we need to understand is why such a complex system with its multiple responses can't defeat HIV. In laboratories throughout the world, scientists are operating at the absolute frontiers of knowledge, or, to put it in another way, at the frontiers of ignorance. Working with the live virus behind the double doors of a security lab, technicians wear masks as well as disposable overalls. They treat HIV as cautiously as the first rocks from the moon. It's hard to get to grips with HIV for several reasons. You can't see it clearly, it goes into hiding, and it's constantly changing. Models of the human immunodeficiency virus do exist, but they're only approximations. Part of the secret of HIV's success is the way its genes operate once inside another cell. When the virus attacks a cell, one of the proteins on the surface of HIV snuggles into a receptor on the surface of the T cell, like a fat man in an armchair. What happens next is like the invasion of the body snatchers. First, the virus injects its genes into the host cell. Then it turns those genes, which are RNA, into DNA. This is the step that most current drug therapies try to interrupt. The viral DNA hides itself right inside the host's DNA, where it can't be seen by the defenses of the immune system. There it stays until a signal prompts it to transform DNA back into RNA and make new versions of itself. Then the new viruses assemble and bud from the surface of the host cell. But many of them have mutated. Their genetic code has changed. The virus is a rather primitive organism and so is bad at making accurate copies of itself. Mistakes can occur when it converts RNA to DNA. That's why the virus eventually wins. The immune system can't cope with a moving target.
Now give me a big swallow, big swallow. Now the tumour's in the esophagus or the gullet.
This AIDS patient is undergoing an endoscopy. Under partial local anaesthetic, a camera is being fed down his throat as far as the stomach to search for infection.
Lymphoma of the esophagus.
It's 8 years since he tested positive for HIV, meaning that he'd developed antibodies against the virus. Since then, though apparently latent, the virus has been slowly replicating. Now he has small green ulcers, probably caused by CMV, a classic AIDS disease. As the virus kills off the T cells, the immune system gradually stops working. Patients fall prey to so-called opportunistic infections and typically develop skin and body cancers. But is this direct killing of T cells all that's happening? The problem with that conclusion is that the virus seems to infect only a small proportion of T cells in the blood, not enough on its own to cause such extensive damage to the immune system. Nevertheless, the T cell is central.
That cell plays a crucial role as, if you will, the conductor of the immunologic orchestra in raising the normal antibody and killer T cell responses. So the immune system is staggered by the AIDS virus because of the quirk that the AIDS virus infects such pivotal central cells in the immunologic response.
We know that in a test tube, the virus can actually directly kill the cell. It infects a cell, it causes it to fuse with other cells and ultimately die in the test tube. Is that the only way that the virus can kill the cell? Unlikely. That's when we get into these indirect mechanisms that I call the gaps in our knowledge of how the virus works.
To me, the great puzzle of how HIV causes AIDS is that we know that HIV can kill the T cells that control the immune system, but we know that the damage in the immune system is occurring often within a few weeks of getting infected, long before there's any evidence of serious long-term cell death. So there must be something else going on.
A new picture is emerging which seems to show how HIV may be causing the death of T cells indirectly, and where in the body it may be happening. new pieces of the puzzle which will have a far-reaching effect on the search for a therapy or even a cure. Amsterdam, host to the 1992 World Conference on AIDS. It should have been held in Harvard, but Boston scientists boycotted that venue because the American government had started to impose immigration restrictions on people with HIV. The Amsterdam conference, the 8th, attracted 10,000 delegates from a veritable AIDS industry looking for a cure, as well as the grants that go towards it. The science and policy sessions were sometimes besieged by pressure groups, so that scientists could be heard complaining that the conference was being reduced to a circus. Gay activists and prostitutes complained that their civil rights were being infringed on account of the spread of AIDS.
Keep your fans low! Forward! Equality! Keep your fans low! Forward! Equality! Rights for whores! Human rights for whores! Human rights for whores! Human rights for whores! Human rights for whores!
For 2 or 3 days, the event was hijacked by a news story about a number of people who had AIDS but didn't have HIV. In a special session, AIDS activists demanded to know why the cases hadn't been reported earlier.
You reported on AIDS in 1981 with a half a dozen cases to a dozen cases. I mean, what difference does that make? I mean, the issue is the issue. Was he AIDS or was he not AIDS?
Today, very few researchers believe the cases are scientifically significant.
The number of HIV-negative cases have been very small, and it's taken us over 8 years to identify this as even a potential problem, and the bottom line is it isn't a problem at the present time, and it mustn't be allowed to distract our attack on HIV-1, the virus which we do know does cause AIDS.
Which leaves mainstream AIDS research carrying on along its painstaking way, searching for the next pieces of the puzzle. Working in Harrow for the Medical Research Council, Dr. Stella Knight's department has been collecting evidence over the last 5 years that incriminates the dendritic cells, named after their dendrites or branches. Those cells are the sentinels of the immune system, lying just beneath the skin. They've developed a method of harvesting dendritic cells from blood and studying them in drops hanging from laboratory dishes. Then they've been able to see how the cells react when they meet the virus.
Dendritic cells are bone marrow-derived cells, which, like other blood cells, and they're then distributed to all the tissues of the body via the blood. And they form, as it were, a network of cells within the tissues. We did try and call them veiled cells because they don't actually have dendrites, they have veils, large, flat flaps that project rather than dendrites. So perhaps veiled would have been a more appropriate description of them. That was the description coined by Dr. Bridget Balfour, who has filmed these cells in action.
It is unclear what the role of that cell is in HIV infection, but some data now, and some of it unfortunately is conflicting at this point, but it'll get sorted out. There is data to indicate that that cell may be the cell that initially has the virus bind to it and then brings the virus to other cells to get infected.
Well, the first evidence we have is that dendritic cells are very susceptible to infection. And if we take cells, dendritic cells, even from asymptomatic individuals, we find that a higher proportion of these cells are infected in many patients than of other cell types, than of lymphocytes or of macrophages. We also see early on a loss in the dendritic cell numbers within the peripheral blood. So there are two things. The dendritic cells are infected and they're reduced in numbers within the blood.
The trail of the dendritic cells leads to the place in the body that is becoming the focus of the new research, the lymph nodes. It's not just blood which circulates in the body, there's also lymph, which is mainly water with salts, fats and proteins, draining from the spaces between the cells. the armpit, and the groin are the main lymph nodes, which filter out any harmful bacteria before sluicing the purified lymph into the bloodstream near the heart. To trigger the immune response, the dendritic cells carry antigen, the foreign bodies, to cells in the lymph nodes.
A lymph node is a small gland that is packed with lymphocytes, which are the effective cells of the immune system. The architecture of the lymph node is such that they have these germinal centers, we call them, which are highly concentrated groups of B lymphocytes that make antibodies. Surrounding that are paracortical areas of T cells that are responsible for helping the B cell or inducing the B cell to make antibody. So under normal circumstances, when you get exposed to an antigen, the antigen is drained to the lymph node. It goes to the germinal center.
One of the things that happens, for instance, when you get a virus infection is you get swollen lymph glands. And this is really indicative of the fact that the antigens are being carried into the lymph nodes and the whole immune mechanism being stimulated in the draining lymph nodes.
The way this happens seems to involve a totally different set of cells, confusingly called the follicular dendritic cells.
In the germinal center is this fascinating complex network of follicular dendritic cells with millions of processes that interdigitate themselves. And each and every lymphocyte in that germinal center, if you look on by an electron microscope, appears to, in one form or another, touch the processes of the follicular dendritic cell. So the follicular dendritic cell is a major antigen-presenting cell because it's right in the follicle of the germinal center. Now, what happens with HIV, that virus gets filtered in the follicular dendritic cell network and just stays there, outside of the cell, coated with antibody. And the cells around there get activated, but also in a unfortunate quirk of the system, instead of just responding to the virus, they are now susceptible to getting infected with the virus. So what we're seeing is the virus is utilizing the primary mechanism whereby the body recognizes and responds to antigen, is using that as a mechanism to infect cells.
The question is, how does it infect them? There's a growing consensus that HIV may be interfering with the communications of the immune system we looked at earlier. Remember how, whenever the immune response is triggered, T cells and B cells are activated by a series of chemical signals. If those signals were altered, it would cause immunological chaos.
The immune system communicates by using signals, cell to cell, and if it's seeing a foreign organism, that organism is presented by another cell to the primary cell of the immune system, and it's through a series of signals that that cell recognizes what's going on, and other specific signals orders other cells to do the attacking, etc. There is growing evidence now that the virus itself may be able to usurp these signalling pathways, i.e. actually set off inappropriate signals and therefore lead to an immune system that is not particularly competent. It's actually sending out the wrong signals to the wrong cells. And this can be done without actually having to invoke any direct killing of the cells by the virus.
The challenge is to understand what the altered signals might be doing. One idea is that they could be causing the cells of the immune system to commit suicide. That idea is not as odd as it sounds, because cell suicide already occurs naturally. Without it, tadpoles would never lose their tails when they grow into frogs, and we would retain the webbed hands and feet we all had as embryos. The same phenomenon occurs in the immune system. It's called apoptosis. Go back to the moment when antigen is presented to a T cell. On the surface of the presenting cell, the antigen is held in a molecule called MHC, shown pink. That's engaged by a receptor called CD4 on the surface of the T cell. When they meet, chemical signals activate the T cell, so, as we know, it multiplies and attacks similar antigens. If these happen to be like some of the body's own antigens, then the T cells will end up attacking the body's own tissues, but they're usually switched off before they can do that.
Results by 3 groups that suggest that interaction between the receptor for the virus, which is a CD4 molecule, and the outer part of the virus, which is the envelope, may induce apoptosis or prime the cell to undergo apoptosis.
There is another possibility, not suicide, but murder. Look at the receptor molecules again, the antigen sitting in its MHC on one cell and the receptor on the surface of the T cell above. What if the virus itself was able to mimic those MHC molecules? What if its protein coat made it look like a foreign MHC molecule? Then this could end up making the T cells of the immune system very confused about what they should be fighting, so they might start attacking healthy immune cells. Well, that's the theory. What's the evidence? Back at the Medical Research Council's lab in Harrow, researchers have made a computer model of the virus's protein coat. They can show that a key part of it really does look like part of an MHC molecule, though computer models aren't accepted as conclusive. If this is how HIV is confusing cell signaling, then it's causing what's known as an autoimmune disease.
An autoimmune disease is a disease where something stimulates the immune system into attacking itself. It feels that it ought to be going after something specific, but can't find it, as it were, and ends up causing damage to the body. A very good example of an autoimmune disease is rheumatoid arthritis, where the damage is all usually limited to the joints.
There are 2 components of autoimmunity as such, vis-a-vis HIV. One is that normally if you get an infected cell, the cell buds a virus off, and your protective surveillance cells that are looking for enemies see this, and in fact this is an enemy, and it kills it. However, the virus can shed proteins, and if the virus sheds a protein that can bind to a cell, this cell isn't infected, it's going around doing its business, but this surveillance cell comes in and sees the viral protein on this normal uninfected cell and is programmed to kill the virus and thinks that this cell is infected with the virus, and boom, it kills an innocent bystander. That's one form of autoimmunity. Another form of autoimmunity is a little bit more complex and not very well understood, and that is that there are components of the virus that cross-react, that are similar in their structure to normal components of the cell, so that when you build up enough immune response against the virus, it may see a component of a normal cell that resembles the virus and inappropriately kill that cell because it thinks it's the virus, even though the virus hasn't gone anywhere near that cell. That is at the stage of being purely hypothetical. There is no data to indicate that that is what is going on in the body in an HIV-infected person.
The trouble is, all these recent ideas are hypothetical, but taken together, they look beguilingly coherent. The virus interferes with the signaling system between the cells of the immune system, somewhere in the lymph nodes, causing them to self-destruct or destroy each other. But we're a long way from proof.
I don't think we can say that at this point in time that one particular mechanism is the predominant mechanism. The phenomena themselves are quite feasible. I mean, a year ago, when there was very little but theory, there was no demonstration that you could actually show this occurring in the test tube. now a lot of those things that were just hypothetical theories are now concretized as being demonstrable in vitro. The question is, what is the in vivo relevance? And we have to be honest and admit that we don't know.
In vivo, in living people, is where research in Baltimore has been focused for the last 8 years. The work's leading to new conclusions which may help to explain why some people's immune systems are apparently able to deal with the virus successfully. The streets around the main hospital make up a deprived neighborhood with a major drug problem, and there's a well-established gay community a few blocks away. Along with several other cities, Baltimore is hosting a major study into local people at risk from HIV. AIDS research so far has concentrated on terminal cases where the immune system is already destroyed. Now there's a growing interest in the early stages of infection. Since 1984, a study at the Johns Hopkins Hospital has been trying to establish what makes some people with HIV live longer than others. The study's control group includes gay men who have no trace of HIV in their bodies at all.
You had any medical problems since we've seen you last?
Not really.
Last 6 months? No?
Not anything that's related to the study.
Have you needed to see your doctor for anything? Good. Taking any medications now?
No, no medications.
Okay. And again? Okay, you can breathe normally now?
On the campus of the National Institutes of Health in Bethesda, blood samples from the Baltimore group have been analyzed at the National Cancer Institute. The team studied the chemical signals of T cells, called cytokines. One of these came from the initial reaction of a T cell to a virus, the killer reaction, known as the cell-mediated response. Then they compared it with another cytokine that triggers the production of antibodies, called the humoral response. They found that as infection progressed, the first cytokine had disappeared and been replaced by the second. In other words, the cell-mediated or killer response had been superseded by the humoral or antibody response. This suggested two things. First, a person's immune system may be more inclined to make one response than the other. And second, the cell-mediated killer response may be more important in containing the disease than the production of antibodies. The balance of these immune responses decides whether or not a person gets sick.
But if the immune system is skewed such that it prefers to do cell-mediated or humoral, what we are suggesting, or at least raising the possibility, is that AIDS investigators consider the possibility that cellular immunity may play an important role, possibly more than antibody, and that the immune system may tend to direct itself in one direction or the other. And the possibility exists that when it directs itself to antibody responses, that it may not be using its whole repertoire of immune function in its most efficient way, so far as HIV is concerned.
The idea of stimulating the cell-mediated and not the antibody response has set scientists on a new track. Chief among them is the man who captured the headlines in 1954 with a vaccine for polio. But the Salk vaccine worked by producing antibodies only, which was effective for a disease like polio. HIV is more difficult because it hides in cells where antibodies cannot reach it. So at present, Dr. Salk is simply aiming to contain the virus after infection by boosting the cell-mediated response, that first response of the immune system to infection, in which the killer T cells are activated.
Now, we have been using virus preparation. It's been purified and inactivated using the whole virus, as distinct from what most other people are doing, and that is they're using genetically engineered parts of the virus. It seemed to me, in this instance, not knowing which component of the virus might be effective, to use it all and not put all our eggs in one basket. And so the principle here is to attempt to bolster the immune system and hopefully prolong its effectiveness, even though the infection exists. it is not expected that we will be able to clear the infection because it already exists in the genetic material of the cells, out of contact with the virus or the immune system. And we're hoping that we can keep the surveillance, the police force, in place so that when virus is expressed, those cells could be destroyed. The idea is to keep the invader, so to speak, at a low level of population.
This moth could hold the key to a different solution, the novel idea of making a decoy for HIV. It's a fall army worm moth, capable of devastating the cotton and cornfields of America. In Boston, the cells of the moth are being used to make a compound which would attract HIV in the bloodstream and immobilize it. The first stage is to culture the moth cells with an insect virus so as to produce CD4, the molecule which acts as a sort of grappling hook for viruses.
And when we use this virus to infect insect cells, they produced enormous amounts of CD4. To tell you that on their surface, they expressed about 12 to 15 times more CD4 than the human T4 cell, the lymphocyte, will make. And once we had this, what could be the best carrier for such a thing if you want it to live for substantial amounts of time in the circulation? Of course, the red blood cell.
The CD4 molecules can be implanted in red blood cells. Because red blood cells have no nuclei, any virus attracted by the CD4 can't replicate. It comes to a dead end. If it proved its worth in clinical trials, the remedy would be specially useful in poor, hot countries. It's cheap, and once dehydrated, doesn't need refrigerating. And it's in poor, hot countries that the need is greatest. Worldwide, 13 million people now have HIV, and in Africa, the numbers are doubling every 2 or 3 years. There are antenatal clinics where 30% of the women are infected, enough to send the population into decline. While in Thailand, prostitution is spreading the epidemic through Bangkok. In one northern Thai province, 15% of army recruits are HIV positive. In India, the same is beginning to happen. Brazil threatens to be next. None of these countries will benefit from the thousands of pounds worth of treatment available in Europe and America. And yet even the best of Western treatment is of little comfort to those with AIDS. It is still a fatal disease, and we have no cure. The main drugs we have now, AZT, DDC, and DDI, all work by stopping viral DNA from replicating. But they only prolong life for perhaps a couple of years. Other possible remedies are some way off, and great gaps in our knowledge of this deadly virus remain. The puzzle of HIV is still incomplete.
So what we really need, and is what we're intensively working on, is the development of drugs that are used in combination, that hit the virus at different points in its life cycle, that can be given early enough in the course of the disease, that are relatively non-toxic, that can truly prolong that disease-free state, so that it isn't only just for an additional year or two, but that it's for 10 or 15 or 20 years. That's the ultimate goal of therapeutic strategy, to do that.
It's frustration. It's frustration. You know what causes the disease. You know the cells it attacks. You've got drugs which can prevent it replicating, a whole range of drugs that can prevent it replicating, and yet apart from controlling the opportunistic infections and the overall care of the patient, which actually has made an enormous difference, and it's really it's extrapolating through into survival, we can't turn the tap off that's causing the disease, and that is total frustration, and that is what we've got to grasp.
I think it's important to realize that tomorrow, or 1 month from now, or 1 year from now, A small observation made in a laboratory, someplace in the world, an observation made concerning how the enzymatic machinery works to replicate the virus, an understanding of exactly what the events are in viral attachment to the cell. Some small observation may prove pivotal in defining a point at which a therapeutic intervention can be made of enormous value. Therefore, we must continue to work to understand as much about the biology of this virus as we possibly can.